Okay, welcome back to where we left off last time, talking about molecular genetics. Just to give it a review of where we left off last time, we finished off with this concept of DNA replication. And if you remember, DNA was a very intricate recipe of all of our proteins, our protein profiles, which basically end up being is a referendum or a representation of who we are. Who we are is really representative based upon our differing protein profiles. So the nucleus of every cell has within it a full copy of our DNA, an incredibly long, intricate molecule. Again, if put end to end, it would be about a molecule of about 6 feet long. And every time a cell divides, that very intricate molecule has to be replicated perfectly, going from one cell into two cells. So thus, that process we went through in detail last time involved a very finely tuned enzyme cascade, which perfectly copies the entire DNA transcript every time a cell divides. We found that this was had a particular challenge associated with it because of the nature of DNA is anti-parallel with respect to the 5' to 3' direction. DNA is replicated in two different directions, and that presented its particular challenges, which had to be met by other enzymes pitching in order to allow DNA to be built or synthesized in two different directions. So really the question becomes, we've talked thus far about how this DNA is information. It's a recipe of everything everything of who we are, every animal or vegetable or life form has within it a DNA transcript. And really the question is, who cares? You can do anything, you can have a whole bunch of information, but what does it actually mean? Well, as I said before, our DNA is a recipe for proteins, for our particular protein profile. But the question becomes, how do these proteins become made? It's very easy to have, say, the plans of a building and architectural plans for a building, but you actually have to build the building. So therein lies a bit of a challenge. So let's see how we can meet that challenge, the actual building of proteins. So this information, which is contained within DNA, has to flow from the DNA and actually build a protein. So here we have just a representation of DNA, which has our information within it. How do we actually synthesize the proteins that are part of that consignment of recipes? Well, just like a recipe book, if we think of a recipe book, has all the recipes within it. And if I just want to make one recipe, I don't need to carry around the entire book with me all the time. I can make a photocopy of one of the pages of that book and allow and just carry out one recipe. Well, our DNA, our entire DNA transcript, can be thought of that book of recipes, and that book of recipes codes for all of the proteins that go into our makeup. Well, if we just want to make one protein, we only need one length of DNA, and that is known as a gene. A gene is a length of DNA that codes for only one particular protein. So we're going to look at this, the synthesis of proteins based upon the building from one gene's worth of information. One gene's worth of information is the recipe for one protein. So let's talk a little bit about that information flow. So we have the information flow from DNA towards the building of a protein. That occurs in a two-step process. The first step in that process involves making a copy. So we're thinking about making a copy. Again, using the analogy of a recipe book, if I have a very large and heavy recipe book, I can, and I only want to make one particular recipe, well, I can photocopy the pertinent page in that recipe book and just carry around that page with me rather than the whole book. This is the process known as transcription. Transcription is making a photocopy. I say that tongue-in-cheek, but it really is a photocopy where we're making a copy of a small amount of information, the information contained in one gene on DNA, and that photocopy involves the synthesis of a duplicate molecule. A duplicate molecule, and we're going to talk about what the structure of that duplicate molecule is, because it transfers that information to the protein building machinery outside of the nucleus. That's very important because the DNA, remember, always stays within the nucleus. Now, the protein building machinery in a cell occurs almost exclusively outside of the nucleus. So the DNA itself, the information in the DNA, can't leave the nucleus, so we have to make a copy of that information to leave the nucleus and go into the cell to build that. So once this transcription process is completed, we have essentially a copy of one gene, and that is sent out into the exterior, outside of the nucleus within the cell, to carry out the actual building of the protein. And this is the process known as translation. Translation is an intricate process where we convert the information on that duplicate molecule, on that photocopy, from one language, that of nitrogenous bases, A, C, T, and G, into another language, that of amino acids. And we know there are 20 amino acids. So this translation process can be thought of as translating from one language into another. So that's where the term comes from. So how does this occur? Well, when we first started this unit of study, we said there were two types of genetic material. We've been talking a lot about DNA thus far, and DNA's really its sole purpose is to stay inside the nucleus of the cell and is the holder of information, the holder of our entire protein profile. Well, when we want to build proteins, we have to look towards what I consider workhorse molecules that actually take care of the bare-knuckled work of building the protein, and that is in the form of the other type of genetic material from DNA, and that is RNA. So, both DNA and RNA are polymers of nucleotides. They're almost identical in that manner, polymers of nucleotides. In nature, we see this concept of the polymer a lot. But there are some notable differences, which we'll take a look at right now. On this figure, we see over on the left-hand side, representation of DNA. And on the right-hand side, we have a comparative representation of RNA. So let's start looking at different structural features, even though, as I said before, they're both polymers of nucleotides. The first structural feature, which starts us right off, it's almost the most obvious, is that DNA is always double-stranded. Two of those linear polymers have to associate together in this in our right-handed double helix, and that's its most stable form. However, RNA is single-stranded, as you can see over on the right-hand side. It is most stable as a single-stranded species. Our next structural difference between RNA and DNA is that with respect to the structural sugar, the monosaccharide that goes into building the two different types of genetic material, DNA's structural sugar is deoxyribose. Indeed, the D in deoxyribose is the D in DNA, whereas RNA, which starts with an R, has a structural sugar ribose, whose structural sugar starts with an R. They're very similar sugars, but they have that slight structural difference. Even so, DNA has exclusively deoxyribose as its structural sugar, and RNA has ribose as its structural sugar. The third structural difference is there are 4 bases that are contained in DNA. They're one-letter abbreviations, C, G, A, and T here. And we have representations of these right here that we see in this representation of DNA. Now in RNA, it's almost exactly the same, and there are 4 bases in RNA structure, and 3 of them are the same as DNA. Indeed, the C, G, and A are the same in DNA and RNA. However, in RNA, we have a very close structural cousin to the thymine in DNA, and that is uracil, given as the one-letter abbreviation U. If we look at these two, so our thymine in DNA looks very much like the uracil in RNA. And if we look very closely, the only structural difference is this methyl group here over on the left on thymine is a lowly hydrogen over here on uracil. So those are our 3 structural differences between RNA and DNA. If we had to look at our differences with respect to its the different tasks, we sort of said this before. DNA is an information storage system. At least the first structure of our RNA, or the first component of our RNA type of structure, is a copy of that information. So let's look, if RNA are the workhorse molecules that do the bare-knuckled work of building proteins, we find that there are 3 classes of RNA molecules. Our first class of the 3 of RNA molecules is known as messenger RNA. This starts out the process. The process of transcription is all about messenger RNA. It is making that photocopy. Messenger RNA is given as this the acronym lowercase m RNA. We find that messenger RNA has exclusively an informational role, which makes sense because if it is a photocopy of the information in DNA, that stays as information in the RNA. It is the copy of one gene within DNA, one protein worth of information. When we talk about one protein worth of information, we are talking about that primary structure of a protein, this amino acid sequence N terminus to C terminus. And it contains the code for, as we said before, that amino acid sequence of a protein. So messenger RNA is an informational role exclusively. We'll look over on the other end of things. Ribosomal RNA. Ribosomal RNA, given as lowercase r, RNA here, in contrast to messenger RNA, has exclusively a structural role. It is the architecture of what is known as the ribosome. A ribosome is a cellular organelle which literally acts as a protein building factory. And ribosomal RNA, along with various other protein substructure, makes up that brick and mortar of that protein making factory. Clearly a structural role exclusively. Our third class of RNA, which may take a little bit more discussion of, is known as transfer RNA, or given as lowercase t RNA as its acronym. It has attributes of both of our previous RNAs. It plays both a structural role and an informational role in protein synthesis. Its structural role is that it literally transfers amino acids to the site of protein synthesis, which we now know is the ribosome. It transfers them, that's where it gets that term transfer RNA, by covalently binding to a particular amino acid and delivering it to the ribosome to be added, that amino acid that could to be added to the growing protein primary sequence. Now, its informational role is really in that second second component of protein synthesis, the translation. It translates from the language of nucleotides into the language of amino acids. So there is its informational role as well. So now that we know our three classes of RNA, let's go right back to the beginning of that protein building complex, that of transcription. We're now tasked with taking the information from one gene, which is ultimately going to be the amino acid sequence of a protein, from the nucleus in the form of a duplicate molecule, this photocopy, if you will. We now know that that photocopy is messenger RNA. Messenger RNA with that informational role exclusively. Now, the building of this photocopy, actually making the photocopy, is that process of transcription. So it is shockingly catalyzed by an enzyme, like most processes within biological systems. And the name of that enzyme is RNA polymerase. RNA polymerase. So let's look at how RNA polymerase both reads the copy of that gene and produces that duplicate molecule. If we look at a rendition of a gene on a protein, we have three sequences which really correspond to three commands that we see over and over in biological processes. These commands are start, continue, and stop. Those three commands. And we'll see towards the end of this lecture other representations of how those three commands are spelled out. But if we look up here on this diagram, we have on the DNA of our gene, we have three components to it. We have a promoter sequence. That's where RNA polymerase binds, and it gives it the command to start. We have the actual amino acid sequence coded into the gene. That is the command continue. And we have a terminator sequence, again in green, which gives the command to stop building the protein. It is complete. So we see within the DNA, we have these three stages. These three stages are given over here on the left as initiation, elongation, that's the continue, and termination, that's the stop. Those three sorts of pieces. So let's see if we can follow this. RNA polymerase binds at the promoter sequence, and down here we see that. There's that sort of that brown figure right there is the representation of RNA polymerase. And if you remember, our DNA double helix structure was very, very stable, and it's very, very difficult to break apart a DNA double helix once formed. We found that in DNA replication that can be done with the enzyme helicase. So it was literally that molecular bulldozer that powered through the hydrogen bonds in that complex and in the double-stranded complex and made it into single strands. RNA polymerase does not completely open up the strands, but we find that the strands do have to be opened somewhat in order for these enzymes to get in there and read what bases there are on the DNA. In a normal double helix, they are pointed towards the inside. The bases are pointed towards the inside of that double helix, so they're inaccessible. What RNA polymerase does is it sort of unravels the DNA just a little bit. It doesn't completely separate the two strands, but we form what's called this open complex. And if you look at the picture here, you see that the DNA is sort of unraveled a little bit. And when it's unraveled a little bit, RNA polymerase can get in there and read the bases on there. Once it is able to read the bases that we see on there, RNA polymerase now starts to move across that gene, reading those bases. And as it reads the bases, let's go down to this next section right here. Here we have an area where RNA polymerase is almost all the way across that gene. As it's reading those bases in that open complex, which you can still see, it is building out that single-stranded messenger RNA duplicate molecule. That is the transcription process. Now finally, when RNA polymerase reaches the terminator portion of the gene way over here, that is the stop command in many processes, including this one, in which RNA polymerase detaches and it releases its completed RNA transcript. That is the messenger RNA. Understand now that RNA polymerase can go right back and rebind at that promoter sequence and move across the gene again, building another RNA transcript. And indeed, we can have multiple messenger RNA transcripts, which can then go into or pass that information along of that one protein. Remember, we're still in the nucleus here, where the DNA resides. Protein building does not happen within the nucleus. It happens outside the nucleus. So these messenger RNA transcripts now are funneled outside of the nucleus, and we take that information, that one tiny bit of information, to the ribosomes, which are outside the nucleus, to build that protein, that one protein. So let's think a little bit about the information that is now contained in this messenger RNA transcript. What it is again, our messenger RNA, or any genetic material, is just a polymer of nucleotides. And those nucleotides now in RNA are A, G, C, and U. Remember, U is substituted for T now that we're in RNA. So we have those four building blocks, those three nucleotides, and here we show them right here. This is a messenger RNA transcript of a whole lot of bases, a whole lot of nitrogenous bases, a whole lot of nucleotides. What do they actually mean? Well, they're actually, they come out, they're really gibberish unless we group them. We group them three at a time into what are known as codons. A codon is a grouping of three nucleotides. And so our messenger RNA sequence is always looked at in groups of three. Codon by codon by codon. So these groups of three codons on the messenger RNA are the actual information, as we'll see later, for translating into the language of amino acids, using in tandem with this with another type of structure, or rather the class of RNA. That would be our transfer RNA, which we'll ultimately look at next. So our messenger RNA transcript is a copy of the information required to build our protein. Now we really start to come to the workhorse molecule that I was talking about before. And this is the transfer RNA, or lowercase tRNA as its acronym, transfer RNA. If we remember, transfer RNA, or any RNA, is single-stranded. And what ultimately happens, our transfer RNA is an 80 nucleotide sequence, 80 nucleotides long, given over in this representation here. If we travel along here, I'm sort of highlighting the boxes within there. We have different boxes within this transfer RNA. Each of these boxes represents a nucleotide. And if you counted all of these boxes here as I'm moving along here, there'd be about 80 of them. They will ultimately, if we get with a single strand of 80 nucleotides, they will collapse upon itself to form what is termed this cloverleaf structure. This representation here could be thought of as a cloverleaf, a three-leaf clover. Here's one of the leaves, another leaf right there. So it is termed a cloverleaf type of structure. But ultimately, what it's for is a dual purpose, and this is really where we get that translation component of our protein synthesis. Remember, our transcription was our messenger RNA. We're making that photocopy. Now the translation, the building of the protein, we have to translate between the language of nucleotides and the language of amino acids. And with respect to transfer RNA, this is where we get this dual purpose, and here is where the structural role of transfer RNA comes in. Looking at this cloverleaf design, I'll bring your attention to the top where we have this hydroxyl right here. We have this OH right on top. That hydroxyl covalently binds to a specific amino acid specific to the whatever transfer RNA we have right here. So if there's 20 amino acids, and there are 20 amino acids, there is at least one specific transfer RNA for each amino acid, and we're going to find that there are actually many more different transfer RNAs than 20, but there's at least one for each amino acid. And what we ultimately have is we have up on top, we have the covalent attachment of the amino acid up on top, and then on the bottom we have our other informational role down on the bottom. So on the bottom, we have something known as the anticodon, an anticodon. Remember in the previous slide, we had talked about the codon, the three nucleotide sequence on the messenger RNA. Well, on the bottom of our transfer RNA, we have what is known as an anticodon. And if we look carefully at the reasoning for this, the anticodon, that end, those three specific nucleotides, plug into the 3-base codon on the messenger RNA. Let's look back to our concept of complementarity when we talk about our 4 bases. Remember, C always associates with G, never any other nucleotide. A always associates with U in RNA parlance, and that is called complementarity. So here, if A always associates with U, notice down here on the bottom of our transfer RNA, AAA. There is only one sequence on the messenger RNA now that anticodon can plug into, would be the codon UUU. Any other sequence and that anticodon will not plug into it. It has to be that particular sequence. So we get some specificity here. Whatever anticodon we have down here is specific for a particular amino acid up here, and that is where the magic of this transfer RNA comes in, for its ability to translate between the language of nucleotides and the language of amino acids, that amino acid covalently bounded to that hydroxyl on the upper end. This slide here shows an actual X-ray crystal structure of what a transfer RNA actually looks like. It doesn't really look like a cloverleaf design. If you look very carefully at this X-ray crystal structure, you could probably pick out the clover leaves, but what they really actually look like is the letter L, or a cowboy boot, or whatever you want to look at that. But it does have 2 specific ends associated with it. Over on the left-hand side, that end is where the amino acid attaches. Indeed, in this X-ray crystal structure, there is an actual amino acid attached to the end, that tiny grouping of atoms that we see right on the very end, right there. That is the amino acid, and what that does is it brings up a happenstance that we see in nature frequently, that we get these very large, very complex molecules tasked with shuttling around very, very small seemingly insignificant molecules. It seems like incredible overkill on the order of nature, yet we see this again and again in nature, these gigantic structures which are tasked with carrying around very, very tiny molecules within biological systems. This transfer RNA is no different. This huge L-shaped structure tasked with only carrying around that tiny squib of collection of atoms, that amino acid, on the other end of it. So we have the amino acid over on the left-hand side, but now over on the right-hand side, that is the area where our anticodon would be. That's the attachment to the messenger RNA that we looked at in the last sequence. So that is all important. Three nitrogenous base or 3 nucleotide sequence of the 80 that go into making up that transfer RNA. It is those 3 that are most important because they plug into the codon on the messenger RNA. So here we have in tabular form what that translation means from the translation of between the language of nitrogenous bases to the language of amino acids. It's called the genetic code. The genetic code is, if we look at, for instance, we see a number of boxes here on this table. There's 64 of them to be precise. And if we look at the box on the upper left-hand side, we see 2 things. We see a nucleotide sequence, U, and we see a name of an amino acid, in this case, phenylalanine. So the UUU, that nucleotide sequence, is the codon on the messenger RNA. On messenger RNA, UUU, those 3 nitrogenous bases, or those 3 nucleotides in a row, means phenylalanine. If we look one box over, UCU, that sequence, that codon on the messenger RNA codes for the amino acid serine. Moving one over, UAU, the codes for tyrosine. UGU codes for cysteine, and so on and so forth. So what we get now is really, this is the crux of it. This is the translation between the language of nucleotides and the language of amino acids, and that is all brought about by the physical process of the transfer RNA carrying the amino acid to the site of protein synthesis. If we look there in the upper right-hand corner, we see a fanciful drawing. This pink ribbon right here is meant to be the transfer RNA. That green ball on the top of it, that is an amino acid, in this case cysteine, and it's really looking at this particular box right here, this box right there, where UGC means cysteine. UGC is the codon on the messenger RNA. Look down here on the bottom of the transfer RNA, AG or ACG. Complementarily, ACG plugs directly into the UGC we see in that box that I highlighted. So, and only those that codon and anticodon will plug into each other. So here now we have this physical connection, really, between the amino acid and the language of nucleotides here on this representation of our amino acid laden transfer RNA. Now, if we think back, we have 20 different amino acids, 20 of them. Now, clearly, there are more boxes on this table than 20. And as I said before, there are 64 to be precise. And that is because more than one codon can code for a particular amino acid. Indeed, look up in the upper left-hand corner of the table again here. Phenylalanine, we have UUU, is phenylalanine, but also UUC is also phenylalanine. looking at the 6 boxes below those, we get 6 boxes or 6 codons which code for leucine, which, it brings up the question, well, is the amino acid leucine more important than the amino acid phenylalanine? We're not here to answer that particular question. It's just that it is so. And in the genetic code, we can have a number of different codons which code for the same amino acid. This means that the genetic code is said to be degenerate over here on the left. Genetic code is degenerate because more than one 3-base codon can code for the same amino acid. Now, importantly, we have 64 boxes here. Every single one of them have meaning. All 64 codons that are possible of 4 possible permutations, how to take 4 nitrogenous bases and put them in all possible permutations comes up with the number 64. Well, 61 of them code for amino acids, sometimes in a degenerate fashion where more than one 3-base codon can code for the same amino acid, but there are 3 others. And here I would hearken us back to or remind us that in nature likes to have 3 commands. Remember, those 3 commands are start, continue, and stop, just like we saw in the action of RNA polymerase, making that messenger RNA. And the genetic code is the same as well. There is, if we look on the genetic code, if we look around this table, I will highlight it here, we have a start codon. A start codon. AUG means start the protein building process. Every single protein must have a starting point. So that command is in every single protein that is synthesized in your body, that start command must be deployed, AUG. Now, interestingly, look below AUG here, we see an amino acid, the amino acid methionine. Now, it's uncertain why nature decided to do this, but the start codon for any protein built within the body also has to also codes for that amino acid methionine. What this means is that every single protein that is built in a ribosome within cells always starts with the amino acid methionine at its N-terminus. Remember, in the parlance of primary structure, we go N-terminus to C-terminus, and the N-terminus always starts us out, always with methionine. So every single protein, when it's built in the ribosome, must start with methionine because of the duality we see here, the connection between the start codon and the amino acid methionine. Now, there is a process in protein before a protein becomes its mature form known as post-translational modification. And building that primary sequence within the ribosome is only the first step on becoming the mature protein. We covalently bind amino acids together, in this case, always having methionine at its N-terminus. One of the most common post-translational modifications is after being built on the ribosome is other processes come in and will almost always snip off that N-terminal methionine for whatever reason. It seems like almost a mistake that nature makes, but almost always that N-terminal methionine will get snipped off for whatever reason. It's just sort of a fluke of nature that we see. So let's review. We have our start codon. That starts our amino acid, or our proteins being built. That start codon, which doubles as the amino acid methionine. 61 of these other boxes are part of that continue command. We're going to continue building this protein amino acid by amino acid until it is finished. And then we have to have that third command, and that is the command stop. And we find that in the parlance of the genetic code, there are 3 stop codons, 3 of them. And it doesn't matter which one is used, these codons on messenger RNA will always end with one of these 3 codons, because that tells the protein building machinery to stop. Notice that of all the 3 of these stop codons, they are not associated with any particular amino acid like we had on our start codon. It was always doubly associated with the amino acid methionine. On our stop codons, there is no such double association. It just means stop. So that we're able to start by starting with methionine, continue throughout any of these structures right here, are putting on amino acids, and finally stop within our those 3 commands that are so important within the genetic code. So let's look at this pictorially. We'll start with our messenger RNA sequence. And again, this is the structure that I showed you a few slides ago, where we can go codon by codon. And these codons have meaning now that we know the genetic code. They're either start codons, codons for a particular amino acid, or stop codons. And let's rearrange this structure just a little bit. We'll group these codons together in order that I can allow this animation. Here's the same thing that we looked at before. I've just grouped the codons a little bit more. Each of these codons will plug in to a particular anticodon, or most of them anyways, except for the codon, into a particular anticodon on the transfer RNAs. So let's see how that ultimately happens. We'll do this all at once. And here again, I've drawn these fanciful drawings in which we have let's see here, our transfer RNAs looking sort of like a sigma right here. It's sort of like we saw in the last one right there. And on the top of these transfer RNAs, we have our amino acids on one end, and on the bottom end, we have our anticodons down here. And each of these plugs in complementarily, and we can follow this along, such that we can see how this goes together. UAC with AUG. Now remember, AUG was our start codon, and UAC, the anticodon on our transfer RNA, always has our methionine attached here. Methionine starts every protein primary structure, at least in theory. It may get snipped off later, as we said before, through a post-translational modification, but it starts us off. That start codon is coupled with the amino acid methionine. And then we just move along. That next codon on our messenger RNA, GGA. If we looked at that tabular form, or that table we looked at before, we would find that GGA is one of the codons for glycine. And indeed, glycine would be covalently attached to a particular transfer RNA, which had, in this case, CCU as its anticodon, plugging directly into the codon GGA on the messenger RNA. And the same thing happens, the next codon, CCC, plugs into the anticodon GGG. That codon, CCC, is one of our proline codons on the genetic code. And finally, we get UUA, which codes for leucine in this case. So you can see that our sequence of events here, or on our messenger RNA going in this direction, lines up perfectly our amino acids in the correct order on the top part of messenger RNA, or the transfer RNA. My apologies. It doesn't necessarily, this is the process. We find that in actuality, building this, it only occurs one transfer RNA at a time. 2 can fit in the ribosome at a time, but we see this as an assembly line. And as we'll remember, is that all-important primary structure determines the ultimate fate and the function of the protein in the end. I should note that this UGA on the end is one of our stop codons, and actually this does not associate with any amino acid. It's a little, it doesn't actually associate with any transfer RNA either. We're going to see specifically how that ultimately works out. It's not transfer RNA that plugs into a stop codon, but rather a release factor. We'll look at that in more detail later, but it means that's that third command given in nature a lot of the times, that of stop. So here we can see from a more artistically sound case the process of going from DNA all the way. So here we up on top we have DNA, and we're going to go all the way to the protein. That's termed polypeptide sometimes. Remember, peptide was a protein of 10 amino acids or less. It doesn't really matter. It just means the protein. So let's look at this process step by step. We have our DNA, and our DNA is two strands put together. One is known as the coding strand, and one is given as the template strand. And from this DNA is what our messenger RNA is built from. Note that the messenger RNA, other than going from the T's to the U's, is an exact copy of the coding strand, but a complementary copy of the template strand, because by definition on our two strands of DNA are complementary copies. So here is our messenger RNA, and again we have our codons written in here, which are sequences of three bases together. From that, we can, and again, this is more artistic than anything else, we have our transfer RNAs all sort of lined up, and we line those up. There are anticodons to codons in each of these cases, and we end up with our particular protein sequence going from start all the way to the end. So, and again, note our start codon always begins with methionine here, although it may be snipped off in the future. So let's bring this home now from a practical protein building sense, the actual synthesis of the proteins. As we saw before, the proteins are carried out, the protein synthesis is carried out on the ribosomes, made primarily from that ribosomal RNA and other complexes of proteins that literally build that brick and mortar of the protein building factory. So here now, within that ribosome, we have moving through it our messenger RNA, which is given right here. These sort of look like field snails, if you will. These are representations of the ribosome, which we'll look at in more detail in just a minute. But the messenger RNA shown here is pulled through the ribosome, and as it's pulled through the ribosome, the ribosome reads the base pairings on it, the codons on it, and builds the protein coming out the other end here. This is the growing primary structures that we're seeing here of these proteins that the messenger RNA codes for. Notice that protein synthesis can occur in multiple places on one messenger RNA transcript at once. Here in this very crude representation, it's much longer than this, but we have the messenger RNA being pulled through 3 ribosomes at the same time, each producing their own copy of or at least primary structure of that protein. So really, we can get many different copies of a protein from one transcript of messenger RNA. So it is said to be catalytic with regards to that. So let's look at this process. Again, we're going to bring in this what nature, the 3 commands that nature likes to put forth: start, continue, and stop. So the start process. We are going to initiate our process here. This is protein translation, the initiation. This is where nature says start. And if we look down here on the bottom, we get our ribosome is made up of two pieces, a small ribosomal subunit, and over here on the right, the large ribosomal subunit, and they squished together sort of like two hamburger buns onto the messenger RNA. We put through there. So we have that messenger RNA being carried through that complex right in the middle, sort of squished between the large ribosomal subunit and the small ribosomal subunit. And what starts us out here is that once we get the ribosome set up, we have two sites on it, the P site and the A site. The P site and the A site. Now the P site we plug in, if you will. We have a transfer RNA that comes in with a particular amino acid already covalently bound to it. That's the one up here. Notice the first thing that the ribosome sees on the messenger RNA, AUG. That's the start codon. It plugs into the anticodon UAC of that transfer RNA, which has on its other end, guess which amino acid? That would be methionine. Remember, every protein starts with methionine, and that starts us off. And then we can move sort of into the continuing process. We're going to be adding an amino acid at a time through this ribosome. So the next, what we're going to see on the next slide is is it somewhere in the middle or close to the end of building this particular protein, at least the amino acid sequence, the primary structure of it. So here we are somewhere in the middle of this protein building complex. This is very representative, if you will, a lot of cartoon here. But ultimately, this is a three-step process, this elongation process right here. We're elongating the primary structure of that protein. So let's catch this sort of somewhere. So over here in the P site, we see our transfer RNA reading a particular codon on the messenger RNA to anticodon on the transfer RNA. And here we have the growing protein chain right here. It is covalently attached at the P site to that green, now given as green, transfer RNA. And a new amino acid laden, or transfer RNA laden with an amino acid, plugs into the empty A site, codon to anticodon. Next thing we see over here is that growing protein chain has been jumped from that transfer RNA in the P site over to the the amino acid on the A site, and a covalent bond is then formed. Subsequently, we have then a shift. This transfer RNA is kicked to the curb, and this other transfer RNA moves from the A site into the P site until we get what we see in this final drawing right here, and that will ultimately start us off again, starting again with another amino acid laden transfer RNA coming in and going through the process again. This cycle repeats over and over and over until the protein has been fully formed. Once the primary structure of the protein has been completed, we're all the way to the end, we encounter a stop codon. If we look over here on the structure on the left, UAG was one of our stop codons, and it does not bind transfer RNA. It binds what's called a release factor, and this given here is this sort of brown Easter eggy looking thing, and that release factor will fit into that stop codon. And once that happens into the now empty A site, once that happens, it gives that third command that we see in nature a lot, stop. Obviously, it's a stop codon, and what that means is the entire complex then breaks apart. The two ribosomal subunits break apart, the release factor moves away, the messenger RNA is released, and most importantly, the finished protein is then released to perhaps fold into its more mature form. Remember, this process, if we were talking about, say, a globular protein, only accounts for the building, the covalent building of the primary structure of the protein. All other secondary structure and tertiary structure, that three-dimensional structure, has to form through different post-translational modifications, and it is not associated with being built at the ribosome. This will finish us off with regards to this molecular genetics unit of study. And I realize it's quite difficult to lecture on how some of these very complex processes are put forth. Thus, I've included here, if you want to look at them, some very good videos on YouTube, which I've found really encapsulate a lot of what we were talking about: DNA replication, DNA folding into chromosomes. Indeed, the process of transcription and translation in these videos is really fantastic. It also helps to open up the fact that we've really just scratched the surface here. We have not even begun to delve into the complexities of what's going on here. Indeed, in at least one of these videos, they'll talk about more about how much we don't know, and the more we discover about these processes, the more strange it seems, and it works towards an understanding of how much we really don't know about the process and how much we're still working towards an understanding of it.